Good morning dear students. നമുക്കിന്ന് ഫംഗയെ പറ്റി പഠിക്കാം. ഫംഗി ഞാൻ അസൈൻമെൻറ്റ് ടോപ്പിക്കായിട്ട് തന്നതാണ്. നിങ്ങൾ +2-നും അതുപോലെ ബോട്ടണിയിലും ഒക്കെ പഠിച്ചിട്ടുള്ളതാണ്. അപ്പം നിങ്ങളുടെ ക്ലാസ്സിലെ തന്നെ കുറച്ച് കുട്ടികൾ അസൈൻമെൻറ്റ് സബ്മിറ്റ് ചെയ്തതിൽ നിന്നും ഞാൻ സ്ലൈഡ്സ് ആക്കിയതാണ്. ചെറിയ മോഡിഫിക്കേഷൻസ് ഞാൻ വരുത്തിയിട്ടുണ്ട് ഫംഗി. അപ്പം ഇത് അസൈൻമെൻറ്റ് ടോപ്പിക് ആയതുകൊണ്ടും നിങ്ങൾ ഓൾറെഡി പഠിച്ചതായതുകൊണ്ടും നമ്മൾ മെയിൻ പോയിൻറ്റ്സ് മാത്രം പറഞ്ഞു പോകും. Fungi are eukaryotes with complex cellular organization. They display two distinct morphological states, the vegetative and reproductive. The main vegetative body of most fungi is made up of fine branching threads called hyphae. Fungi with vast numbers of hyphae, all interconnected to make up a tangled web called mycelium. Most fungal hyphae are divided into separate cells by end walls called septa. The fungal cell contain a membrane-bound nucleus where the DNA is wrapped around histone protein. The rigid layers of fungal cell wall contain complex polysaccharides called chitin and glucan. So this is about the cellular organization of the fungi. It exists in two different stages, that is its reproductive stage and its vegetative stage. And the vegetative state, the fungal body is called hyphae, and it is a fine thread-like structure. And the hyphae, the large number of hyphae, they get interconnected to form a web-like structure called mycelium. Then fungal hyphae, it is divided into separate cells by means of septa, and some septa may contain more than two nuclei or more than one nuclei. Then they are called cenocytic hyphae. And the fungal cell, it is we have already mentioned that it is a eukaryote, so it is having a true nucleus, so membrane bound nucleus is present, and the DNA, it is wrapped around the histone protein. Then rigid layers of fungal cell wall contain complex polysaccharides called chitin. Chitin is also present in insect exoskeleton, so chitin, along with the chitin, the fungal cell wall contain glucan. So fungi is an organism which is possessing cell wall. So bacteria, plant and fungi possess cell wall. Next is reproduction. Reproduction may be vegetative, asexual or sexual, and vegetative reproduction occurs either by fission, fragmentation or by budding. And sexual reproduction, it involves sporogenesis, plasmogamy, karyogamy and meiosis. In both sexual and asexual reproduction, fungi produce spores that get dispersed from the parent organism either by wind or through animals. Asexual reproduction, it involves chlamydospores and conidia. So asexual reproduction also involves the formation of spores. It is either chlamydospores or conidia. Next is nutrition. Nutrition is heterotrophic and absorptive type. Plastids, chlorophyll and other photosynthetic pigments are absent, so fungi are non-phototropic organisms. Mycology is the study of fungi, and most fungi are saprophytes. That means they are decomposing organisms which live on dead and decaying material. Next is classification of fungi. Kingdom fungi or mycota, it is divided into three divisions, namely gymnomycota, mastigomycota and emastigomycota. So kingdom fungi, it is mainly divided into three main divisions: gymnomycota, mastigomycota and emastigomycota. Division gymnomycota, it includes phagotropic organism devoid of cell wall. This division comprises of two subdivisions, namely Acrasiogymnomycotina and Plasmodiogymnomycotina. So division gymnomycotina, it is composed of fungi without any cell wall, and it is further divided into two subdivisions, namely Acrasiogymnomycotina and Plasmodiogymnomycotina. Second division is Mastigomycota. It includes fungi with absorptive nutrition, unicellular or filamentous fungi with a cenocytic mycelium. That means mycelium is composed of more than one nuclei in a single cell. It comprises of two subdivisions, namely Haplomastigomycotina and Diplomastigomycotina. Division Mastigomycota, It is composed of fungi with absorptive nutrition. These fungi are unicellular or filamentous, and their mycelium might be cenocytic. And it is again divided into two subdivisions, haplomastigomycotina and diplomastigomycotina. Then the third division, it is amastigomycota. Here this fungi is also with having absorptive nutrition. They are having mycelia aseptate or septate, and the cells are non-motile, and this include four classes, namely Phycomyces, Ascomyces, Basidiomyces and Deuteromyces. So division Amastigomycota, these fungi are also having absorptive nutrition with a septate or aseptate mycelia, and four main classes coming under Amastigomycota are class Phycomyces, Ascomyces, Basidiomyces and Deuteromyces. First class is class Phycomyces. They are found in aquatic habitat. Mycelium is septate and cenocytic. Asexual reproduction takes place by zoospores or aplanospores. The zoospore is formed by the fusion of two gametes. The gametes are similar in morphology, and examples include mucor and rhizopus. So Phycomyces, they are aquatic fungus having septate mycelia and cenocytic mycelia. Reproduction is by asexual mode, and it is by producing zoospores and or aplanophores, and zygospore. Sexual reproduction involves the formation of zygospore, and this zygospore is formed by the fusion of gametes. The gametes are similar in morphology, and example include the common bread mold, that is the rhizopus and mucor. And next class is class Ascomyces. It is known as sac fungi. They are multicellular, saprophytic, decomposers, or they are parasitic or coprophilous. Coprophilous means they eat corpus, that is the fecal material of other organism. Then asexual spores are conidia. Sexual spores are called ascospores. Many members in this group are edible. Example Aspergillus and Penicillium. So we have both these genus are familiar to us. So the Ascomyces is otherwise known as sac fungi. They are multicellular, and they are saprophytic, decomposers or parasitic, coprophilous fungi. They have both sexual and asexual mode of reproduction. Sexual is by means of ascospores, and asexual by means of conidia. Most members are edible. Example Aspergillus and Penicillium. Next class is Basidiomyces. Class Basidiomyces is commonly known form of Basidiomyces are called mushrooms, bracket fungi or puffballs. They grow in soil or logs, on tree trunks, and in living plant bodies as parasites. Rust and smuts are example of Basidiomyces. Sex organs are absent, but plasmogamy is brought about by the fusion of two vegetative or somatic cells of different strains or genotype. Resultant structure is called basidium. So here sexual reproduction is absent, but plasmogamy is there, and in plasmogamy either somatic cell or vegetative cells of two different strains or genotype get fused to form a structure called basidium. And this Basidiomyces include mushroom, bracket fungi, puffball, etc. And they are seen either in soil or in tree stems or in plant bodies. They are mostly parasite and include common examples are rust and smuts. So their reproductive structure is called basidium. Okay. So that is all about Basidiomyces. Then the last class is class Deuteromyces. They are commonly called fungi imperfecta because only asexual or vegetative phase are known. So their sexual mode of reproduction is quite unknown, and they reproduce sexually by means of conidia. Mycelia is septate and branched. Some members are saprophyte or parasite, while large numbers of them are decomposers of litter, and they help in mineral cycling. Examples are Alternaria and Trichoderma. So Dicteriomyces is called fungi imperfecta because of the absence of knowledge about their sexual phase. Sexual phase. A sexual phase is by means of conidia. Here the mycelium is septate as well as branch, and they are either saprophyte or parasite or decomposers, which helps in nutrient recycling, and example include Alternaria and Trichoderma. The conventional classification of fungi is over. Now, based on morphology, fungi can be classified into 4 different groups. Yeast, they are eukaryotic unicellular fungi. Some yeast are dimorphic, that means they can exist in 2 different forms. One form is the normal oval budding yeast, and other form is, in certain conditions, this budding yeast, it will form a filamentous like structure called hyphae, similar to that of other mold. Next is yeast-like fungi. Here the fungi which occur in the form of budding yeast-like cells and as chains of elongated unbranched hyphae. So here yeast is like that of a budded yeast, and it forms chains, but the chains is elongated with the unbranched hyphae. The hyphae then intertwined or that interconnected to form pseudomycelium. The yeast like fungi are grouped together in the genus Candida. So Candida is a yeast like fungi. Candida, it is a genus of yeast, and many species are harmless commensals or endosymbionts of humans, but some species can cause disease. Candida albicans, which we have already studied, is an oval budding yeast. It will cause candidiasis in humans, and we have already studied different form of candidiasis, and it is a diploid fungus that grow both as yeast and filamentous cells. So it is a diploid one or dimorphic one, which can grow both as a normal yeast and as well as a filamentous cell. Next is filamentous fungi. Filamentous fungi are of critical importance because they can produce certain commercially important enzymes and organic compounds. They are mostly saprophytic organisms which secrete a wide variety of enzymes which are involved in the decomposition as well as recycling of complex biopolymers from both plants and animal tissues. So the filamentous fungi is having a commercial importance because of the presence of enzymes and other organic compounds present in them, which can be used for the decomposition of decomposition of organic materials, and also help in the recycling of complex biopolymers of both plants and animal tissue. Dimorphic fungi. It is a fungi that exists in the form of both yeast and mold. That means dimorphic fungi can exist either as yeast or as filamentous form. Several species of dimorphic fungi are important pathogens of humans and other animals. Example is Talaromyces marnefi. Talaromyces marnefi. So it is a dimorphic fungi which can exist in the form of both yeast and filamentous fungi. So the last one is importance of fungi. Fungi are mostly saprops. That means they are saprophytic organisms which can derive nutrients from decaying organic matter. Fungal exoenzymes are able to break down insoluble polysaccharides such as cellulose and lignin of dead wood into readily absorbable glucose molecules. It also helps in the recycling of carbon, nitrogen and other elements into the environment. And fungi are can be used as a potential tool in bioremediation process. That means fungi have the capacity to eliminate pollutants from the environment. So fungi, their importance are they are saprophytic organisms. They produce certain commercial enzymes which are used for breaking down the complex materials that are present in plants as well as animals, and it helps in the recycling of nutrients like carbon, nitrogen and other elements in the environment, and it can be used as a potential tool in bioremediation process. So that's all about fungi. Thank you.